The potential for the survival of life signs exists in close proximity to the surfaces of both Enceladus and Europa. Greetings, everyone. Today, we will be delving into the fascinating subject of the survival of amino acids, which are considered as potential indicators of life in the vicinity of icy moons such as Enceladus and Europa. Recent research has revealed that these amino acids can endure radiation and degradation under specific circumstances, eliminating the need for extensive drilling to detect them beneath the surface. This significant discovery further strengthens the notion that the subsurface oceans of these moons may have the capacity to support life. It also emphasizes the utmost importance of careful sampling during future missions to these captivating celestial bodies. Without any further delay, let us embark on our insightful discussion. According to Alexander Pavlov, the lead author of a research paper published in Astrobiology, our experiments have determined that the optimal sampling depth for amino acids on Europa is approximately 8 inches, around 20 centimeters, in the high latitudes of the trailing hemisphere. This refers to the hemisphere opposite to the direction of Europa's motion around Jupiter, specifically in areas where the surface has not been significantly impacted by meteorites. On the other hand, detecting amino acids on Enceladus does not require subsurface sampling. These molecules can withstand radiolysis, or breakdown by radiation, at any location on the surface of Enceladus that is less than 1/10 of an inch, a few millimeters deep. The icy exteriors of these airless moons, which are devoid of warmth, are probably unsuitable for sustaining life due to radiation emitted by the host planet's magnetic fields and cosmic events like supernovae. Nevertheless, beneath their frozen crusts lie vast oceans that are warmed by the gravitational pull of the host planet and nearby moons. If these subterranean seas possess the essential components for life, including an energy source and the necessary elements and compounds for biological processes, they could potentially be home to living organisms. In their radiolysis experiments, the research team explored the possibility of using amino acids as representatives of biomolecules found on icy moons. Amino acids can arise from both biological and non-biological processes. However, the discovery of specific types of amino acids on moons like Europa or Enceladus could potentially indicate the presence of life, as these molecules are crucial building blocks for proteins in terrestrial organisms. Proteins play a vital role in life by serving as enzymes that accelerate or regulate chemical reactions, as well as forming structural components. It is plausible that amino acids and other compounds from subsurface oceans may be transported to the surface through either geyser activity or the gradual movement of the icy crust. In order to assess the viability of amino acids on these celestial bodies, the researchers combined samples of amino acids with extremely cold ice, reaching temperatures as low as -321 Fahrenheit, -196 Celsius, within sealed vials devoid of air. These samples were then subjected to varying doses of gamma rays, a form of high-energy light. Additionally, to account for the possibility of microscopic life existing in the oceans, the team also examined the survival of amino acids within deceased bacteria that were frozen in ice. Lastly, they investigated the behavior of amino acids when mixed with silicate dust in ice, considering the potential interaction between materials from meteorites or the planet's interior and the surface ice. By conducting experiments, the team gathered crucial information regarding the radiolysis constants, which are responsible for determining the rate of amino acid degradation. Utilizing this data, along with the knowledge of the ice surface's age and the radiation conditions on Europa and Enceladus, the team was able to calculate the specific depth and areas where 10% of the amino acids would remain intact, despite radiolytic destruction. Previous studies have conducted experiments to examine the viability of amino acids in ice, but this particular study stands out as the first to utilize lower levels of radiation that do not completely disintegrate the amino acids. The purpose of this approach is to avoid complete destruction and instead focus on altering or degrading the amino acids, as this allows for a more accurate assessment of their potential as indicators of life. Additionally, this study marks the first instance of replicating the conditions found on Europa and Enceladus to assess the survival of these compounds in microorganisms. Furthermore, it is also the inaugural experiment to investigate the endurance of amino acids when mixed with dust. The research team discovered that the degradation of amino acids occurred at an accelerated rate when combined with dust, whereas the process was notably slower when originating from microorganisms. Pavlov emphasized the significance of the sluggish degradation of amino acids in biological samples under conditions resembling those found on Europa and Enceladus. This finding serves to strengthen the argument for conducting life detection experiments during future missions to these moons. It is important to note that our research indicates a higher rate of degradation for potential organic biomolecules in silica-rich areas on Europa and Enceladus compared to pure ice. As a result, caution should be exercised when collecting samples from silica-rich regions on these icy moons during potential future missions. One possible reason why amino acids endured for an extended period within bacteria relates to the impact of ionizing radiation on molecular structures. This radiation can directly disrupt chemical bonds or indirectly generate reactive substances in close proximity, which in turn can modify or degrade the targeted molecule. It is plausible that the cellular components of bacteria provided a safeguard for amino acids against the reactive compounds generated by the radiation. The research received backing from multiple sources, including NASA under award number 80 GSFC 21M0002, the Fundamental Laboratory Research Work Package at Goddard through NASA's Planetary Science Division Internal Scientist Funding Program, and the NASA Astrobiology NFOLD Award 80 NSSC 18K1140. To support our channel's growth and ensure broader awareness, kindly hit the like, share and subscribe buttons. This will help us reach more individuals and disseminate valuable information. Thank you in advance.